Thank you very much. Hi, I'm Roger Packer. I'm a pediatric neurologist and head of the Brain Tumor Institute at Children's National. As we talk about the era before there were no cell phones and other things, I was born in that era. I didn't think it was that backwards then, but in retrospect, it was a different time. We actually had phone booths and other things that one day, if you wanted to go to a museum, you could probably see them. But we did our best at that time of trying to help these children. And it's been a long battle. And as I'll show you, we've made strides, but there's so much more to do. And I think this first slide saying that our patients have no time to lose, all patients have no time to lose. And if we lose this battle for a child with medulloblastoma, we lose the child. And if we use therapies that we have now available, we risk the chance that we will cause some degrees of permanent disabilities for the child. And that is the dilemma. And we just heard from Fernando, the other reality of this is with very few exceptions, but important exceptions. If the tumor does come back, we are not able to effectively treat it. Medulloblastoma is the most common high-grade pediatric brain tumor. It is not a disease just in the United States. It's a worldwide disease, and it is one that is devastating, and at this point requires treatment with surgery, and for most patients, radiation and chemotherapy. And as we've talked about, there's no established treatment for relapse. Much, however, that we'll talk about for medulloblastoma, that the discoveries are generalizable, as we'll talk about, and we may be able to help other children with brain tumors. And right now, the unfortunate truth is that childhood brain tumors are the most common form of malignancy children will have, and is also the one with the highest morbidity. It will kill the most children. So we're dealing with a very devastating illness of which we've made some strides, but for other things we haven't gotten where we want to be. And this whole issue of survivors facing lifelong impairments, and these impairments, to just really say what they are, these are abilities to learn in school. These may be abilities to socially function, to, if we can have them beat this disease, to raise families, to have children, to be a normal, whatever that is, member of society, can be limited by the therapies we use. And we've been trying very hard to refine those therapies. And the treatment protocols, and I was involved in the development of those protocols, and for better or for worse, my name is linked to those protocols. They really did make a difference to raising survival rate for some subtypes of medulloblastoma, as high as 80% of children being alive 5 years and maybe cured of the disease. But there were other subtypes of this disease where we really couldn't change the prognosis, and less than half of the children would succumb to disease within 3 to 5 years from diagnosis. And the tragedy, and obviously in a young child and a family and having a child diagnosed usually between 4 and 9 years of age, and what the impact is on the child and the family, gives us all pause in wanting to do more and do things quickly. So one of the challenges that we have is that there is some government funding, not very much, to try to support some of these work. But they really try to aim at maybe the more common brain tumors. Actually, ones that you've already made some progress in, because they can see the progress immediately. And the science doesn't always allow you to do rapid breakthroughs. They are very good at refining things, but if we want to make rapid changes, oftentimes those grant funds are not adequate. And you can only get grant funds once you've already made the discovery to further refine it. And as Fernando said, we wanted to push the envelope and make these discoveries quicker. It usually takes at least 10 years to go from discovery to where we can give the medicine to the child or the therapy to the child. And we knew that during those 10 years, we were going to lose more and more children. So although Fernando states that he was surprised by the rapidity, he told me when we started this that he wanted us to have an answer in 2 years. And it wasn't a surprise to me. And we had to get together a group of institutions that would be willing to work on this problem in concert together to make the timeline to go from idea and experiment and investigation to clinical care in 2 years. He actually asked for 1 year, which I said I couldn't deliver. But we thought in between 2 and 3 years, if we changed the way we work and knowing what I knew of the disease and what some people were doing around the world, maybe we could make that work. And the one group that he wanted us to focus on was one of the groups that scientists were not working on because it was the most difficult form of medulloblastoma model. Medulloblastoma is now broken into multiple different groups, but they're essentially, depending on you count, 3 or 4 groups, and the fours are the largest group, and the one where we still have the least understanding of exactly where it came from, although that work is now breaking because of work of great scientists like Michael Taylor now in Texas, and Dr. Northcott um, in St. Jude's. And we knew that we had an opportunity because things were breaking in this area that we could move things quicker and maybe get to therapy. And that's Dr. Singh, who's one of the investigators we asked to help us, and she from McMaster Institute in Toronto, and was the first one to successfully model group 4 in the lab so we'd have something to test. But unfortunately, her model hasn't been easy to use and has not given us all the insights we wanted. Next, please. So this was the approach that Fernando brought to us from Brazil. We promised to gather the world's experts. We promised no silos. We would not duplicate work. We would not go to labs that never worked in medulloblastoma, although that sounds exciting. It would take them 3 to 4 years to retool their laboratories to really get expertise. We wanted the scientists who knew the disease, were already working maybe on different subtypes. And the question is, could you, with the right enough funding, take part of your laboratory effort, move it towards the group 4, and work as a team, which is not always done in science, with goals. And what attracted the scientists is we said that, our goal was not to have your work get translated into a therapy in 10 years. Our goal was to get it to translate to therapy in 2 years, which they all thought was a little bit nuts, but did agree that they would give that opportunity a chance because that's what they wanted to see. They wanted their research to be meaningful, and we've learned anything from scientists. They do want their work to be meaningful. And we want to accelerate and not duplicate it. Can I have the next slide, please? So beginning with about 6 or 7 laboratories, and now because of the support that people around on this Zoom and Fernando has given us, we've been able to really get multiple centers. We're up to about 13 laboratories throughout the United States and Canada. We are also working with the leading medulloblastoma center in Germany at Heidelberg University. If you look at some of those institutions, they are some of the most important and successful institutions in the world. And we also have a very strong foothold with Dr. Eppelman in Brazil. And our goal is to develop a kind of therapy that would not only be used in children's in Washington, D.C., or the University of California at San Francisco with the group there, or Texas, but be able to transmit this therapy across the world. And the reason that all of these groups signed on was that they were interested in the problem, they had expertise in the problem, and they all were people that I guess I knew quite well would follow the rules of sharing their information immediately, which we do, so that we could all benefit from it and move the condition even faster, farther. Next, please. And this is the list of now the 14 laboratories and the 18 scientific leaders. And we did pull from some areas that really had different expertise. Taking a look at this list, Dr. Weshla Rea from Columbia University was a world expert in developing models and understanding the biology of the tumor. Dr. Mitchells and Sayur from University of Florida were leading the charge internationally, developing immunotherapies, vaccines, CAR T therapies, things to allow the body to fight its own tumor and give it back cells and vaccine to stimulate it to fight its own tumor. And I think you've read a lot about the potential benefits of this. It is a tremendously powerful technique if we can harness it. Dr. Taylor, Texas Children's, has identified, he believes, the cell finally that medulloblastoma comes from. To show you how backwards we were, we're still calling this medulloblastoma, meaning an abnormal growth of medulloblast cells. The only problem, there's no such thing in the body as a medulloblastoma cell. So we really don't even have the right name for it. We have people like Dr. Ramaswamy at SickKids in Toronto and his team doing a great deal of the work in the molecular biology of the tumor and developing new therapies. Dr. McDonald at Emory University looking at new drugs. And as we said, we took the patient, the leading doctors in Germany, who's done a great deal of work in harnessing the genetics of these tumors and letting us understand these tumors. All brain tumors are working on our team. Plus a host of other people from our laboratories at Children's National in Washington, D.C., trying to make a difference, including Dr. Haidar, who's developing the first true CAR T cell to attack the group 4 medulloblastoma, Dr. Kochman, who's doing some drug testing for us to make sure the drugs we're using get into the brain, and Dr. Weiss from University of California, San Francisco, who really has been a leader internationally in developing different molecular ideas about different kinds of brain tumors, including medulloblastoma, but had never worked with group 4. And we asked them all to try to work together to take parts of their laboratories or hire people if they had to with the funding and really try to make a difference working together. And then we wanted to be sure that this information was getting to Brazil and would work with the Brazilian group carefully with Dr. Eppelman in Sao Paulo to make sure that we were developing the kinds of therapies that would not only be available in children's in Washington, D.C., but would be really available internationally. So these are some of the people we pulled together. And for everyone I show you on this list, there's others that they're collaborating with their institution to try to make this very audacious or hard goal something that's a reality. Can I have the next slide, please? So what have we done? Well, when we started this, we thought that the low-hanging fruit for this would be the molecular genetics of the tumor. And with all of this expertise, we would find a drug through all of our screenings that would be the golden bullet that would attack medulloblastoma and destroy it. We would develop the cell lines that would allow us to test it. The frustrating thing, and this is where the reality of science is, if we had put all of our efforts into those two areas, we would have little to show for it yet. That's why we wanted to encompass both the immunologic issues and the molecular therapy. Now, we haven't given up on that, and there are a couple of cell lines that are working. And one of the things, I don't know if you've heard the term organoids, but these are ways to sort of train cells and tumor cells to actually become like organs, like the brain. And this is and our researchers have started moving into organoid work with some of their funding to try to develop mini brains from the medulloblastoma tumors that we can test some new novel therapies again. So we haven't given up on human cell line or the low-hanging fruit, which wasn't so low-hanging. But what we have been able to do in sort of record time is get two FDA, that's the Food and Drug Administration, which is our sort of referees in the United States, approval for two clinical trials, not in 10 years, but within 2 years. Both of the trials have been approved, and one is about, and I'll explain a little bit more about what they are. One is about to enter patients for the first time in the next month or so, and another, once there's a little bit more safety data in one other tumor type, will be able to develop the first vaccine trial for children with medulloblastoma. Again, a trial that up to recently, because of the technology involved, would take 7 years at a minimum. And this has only been doable because of the great sustained giving and focus of our donors to try to make this happen. And we certainly know in the future, as these trials move along, we will be getting government funding and other funding to perform them once we show that it can be done. But no one funds you until you've already shown it can be done, at least in a group of children. Next, please. So the first trial is called the match point trial, where we're training the child's own cells to seek and destroy its medulloblastoma. And for the first time, we're using the tumor's defense against it. The tumor tries to make the immune cells not as effective, and we're adding an additional drug to overcome that, called a checkpoint inhibitor. And I think we have a cartoon on the next slide a little bit more about this. But the idea is make cells that will destroy the tumor and not hurt other cells in the body. The second trial, which is really even more space age, if you remember that the issue with COVID is that nobody had a vaccine or shots against COVID, and the virologists told us, using the old technology, it takes 3 to 4 years to make a COVID vaccine. Very smart people, many of them in industry, some of them with government funding, realized you could do a vaccine differently and make that vaccine and create it in months. And that's how the COVID vaccine was created. We wanted to ask, why couldn't we do the same thing for medulloblastoma? Why couldn't we use some of the building blocks of the genetics of the medulloblastoma to make this vaccine, just as we did against COVID, designed to destroy the tumors? And the answer is, I think we can, and we have. Our team has. Next, please. So let's go back one slide. I apologize. So we didn't put in the cartoon, but these things are intricate. What we're asking is, for the first two studies, we actually take tumor tissue out, and we either make these expanded cells in our laboratory and give them back to the child, or we create a vaccine, and with different medications to take the blocking from the things that block the cells from working, the checkpoint inhibitors, we give these medications back so the immune system soups up. Now, the one risk of doing this is, as we soup up the immune system, remember the immune system can attack the child's normal cells. And what we have to be is very selective that what we're creating are vaccines or immune cells that will only attack the tumor. And we have to do tremendously careful testing in these children. And what we've agreed upon as a consortium is that we will do this preliminary testing with our institutions. No one's going to make a profit off of this. We're going to try to do the first 10 or 20 children. And then if we are able to do this appropriately, we will hopefully be able to then work with larger consortiums and get this to more children more quickly. Next, please. So this is what we have. We have the immunotherapies. What's going to come next? Where there are still laboratories working for this magic bullet that will kill the cells. So drug development testing is now in its third year, and there's some good hints of where we might be able to go and maybe combine it with immunotherapy. A new science opened up at the same time we were doing this, the discovery of what is the origin of group 4. And Dr. Taylor, who's working with our group, and especially with some of the monies that were supporting him, was able to isolate where the group 4's medullary tumors were, which were really in infancy, in little rests in the back of the brain, with areas that seemed to be sort of inactive, and then they group into medulloblastoma. And we're going to use that information, hopefully, to even make better vaccines. And Dr. Taylor's hope is that he can figure out a way to use that information to prevent medulloblastoma from ever happening. The CAR T cells, the magic bullet, where the lymph cells are loaded with payload, the lymphatic cells are loaded with payload to get in and attack the tumor. They have not worked in other kinds of childhood brain tumors yet. We have Dr. Haydar and a couple of others in our group making selective CAR T cells for the group 4, and we think that's going to be ready for therapy in the next 2 years. We have created what we call a liquid biopsy, a platform where we can monitor in blood whether therapy is working quickly or not, whether we have stimulated the immune system appropriately or not. That's going to be incorporated into our trials. And all of this will be what we hope is a series of trials. We hope our first 2 trials will be home runs, and we don't need anything else. Unfortunately, we cannot rely on that. And we have to have, if step A works only for some, how do we add to it or go in a different direction so it'll work for more? And we also have the reality that what we are dealing with is not something for patients in 10 years, but what can we do now and in the immediate future? So that's what's next for that group. And none of this happens without the philanthropy of Fernando and all of those on the call here. And from the bottom of our hearts and the bottom of the hearts of our researchers that really want to make a difference, we thank you tremendously for your help and your trust. And we would really like to take this to the next level and start curing children.
So the first question would be, Dr. Packer, if someone on this call is interested in getting more recommendations or more information on recommendations on how to treat their child, they're dealing with some specific things that they would like you and your team's advice on, what's the best way for them to get connected to you and your team at Children's National?
Yeah, there's a couple of ways, but I would like to make the first statement that we are doing our best to get the information generalizable. So we believe the first thing is always to talk to your team taking care of you at that institution. They're probably working extremely hard to look for different answers and really know you the best. But if we can play a role as a second opinion, I'm sure Andrew at the end of this will get some contact information to everybody on the Zoom. And there's a couple of ways. If it's a very specific question, I or one of my team are happy to take the call. If it's more complex and has to do this right off the time, we have to review all the records. We have to look at lots of MRI scans. If we give you a knee-jerk response, we're probably going to be wrong half of the time we try to do it, and we can't afford to be wrong. So we do have a mechanism for both getting you here in person to do a second opinion, which is always the best, or to do a remote second opinion if we can gather the information, and then we can either, after the information is there, either at your wants or whatever you'd like to do, we could either then meet in person by Zoom, or we could prepare a written record back to your physician. And there's a remote second opinion team that I'm sure Andrew can get a hold of with Camille in my office of how you make that contact. I also have an email address. You can always email me. He'll send that out. The problem there is that if I really don't know the situation, I really probably won't be able to give you a detailed enough information or reply to be meaningful for your child. So yes, we do second opinions. We do remote second opinions. As far as where the studies that we talked about are presently available, right now, if we believe someone is a candidate, the first 2 patients will be done at the University of Florida with Dr. Mitchell's team, because that's the FDA wants the people who make the therapy to do it first at their site. And then we hope in the next few months, we'll have these things available at our site. Similarly with the COVID vaccine, the first one, the COVID-like vaccine that uses the RNA technology, the first will be done at Florida. And then after that, it'll be done here. And we will probably then open it up to 3 or 4 other sites if they have the capabilities of handling vaccine trials.
And then the other question for Dr. Packer, and then there's one for Fernando, would be when we look at the consortium, there were a few questions on group 4 and what that means and how that relates to the specific research being done by the consortium. Can you just touch a little bit more regarding the focus on group 4?
I wasn't going to try to get into completely the weeds of this, but essentially,
Sure.
Even though medulloblastoma itself is the most common single malignant brain tumor we deal with, and that's all we really knew until about 10, 12 years ago. Now, understanding the makeup of these tumors, the tumor has been split into as many as 17 different subtypes. And of those, although some of them share things, there are 3 or 4 major subtypes. And they're driven by different genetic mutations. And even though we do great with some kids with one of the mutations, aiming in a therapy at the time of recurrence at that mutation makes no sense because the child might not even have it. So the most common subtype is the group 4, which makes up about 40 to 45% of all of the medulloblastomas. Even within group 4, and now is merged into group 3, there are 8 different subtypes. So each one has a different molecular makeup. Chemotherapy, when we used it in radiation therapy, was sort of, and were agnostics. They didn't care. They blasted every dividing cell. That had its pluses and minuses. You'd kill normal cells, but you'd also kill medulloblastoma cells. Immunotherapy and molecular targeted therapy have to aim at a specific target on the cell or in the cell. And that's where it's very important. If we're going to try to treat children with this subtype, we have to have an immunotherapy or molecular therapy which is personalized to that child. And that's what we're trying to do. And of those, the orphan disease in medulloblastoma was group 4 because nobody really understood how it developed. What did it do? And that's why it was so important to start this initiative and tell people, go ahead and work on the other subtypes that you understand a little bit better. But how about focusing on the one you don't understand, that you don't know what the drivers are, the genetics are. And now we know better what the drivers and the genetics are. And really that's breaking out for work that's going on in these laboratories and other laboratories around the world at sort of a record pace that we didn't know anything, and now we know a lot, but we don't know how to cure many of them yet.